Hello students, welcome to chapter 11. We're going to be talking about meiosis. This is different from mitosis, where the cells are duplicating in copies. Meiosis, this is where cells are duplicating in basically in half. All right, and I'll show you how that is happening. So in meiosis, the cells are duplicating basically through the process of producing gametes. Okay, we all know gametes are the eggs and sperm. So we have the males produce sperm and females produce eggs, right? So these are dependent on meiosis. So gametes have half the number of chromosomes present in the somatic cells or the body cells. So all of the rest of our cells are somatic cells, except for eggs and sperm cells. So we have our regular cells that have 46 chromosomes, or 23 pairs of chromosomes, and then the sperm and egg cells only have 23 chromosomes. At fertilization, the nuclei of an egg and sperm fuse together, right? So we have one sperm and one egg. That's 23 and 23. They fuse together, and then you get 46. The making of gametes is called gametogenesis. This is the cellular mechanism of gamete formation. The two representatives of each chromosome in a diploid cell constitute a homologous pair. Homologous just means that they have the same genetics. They have the same genes arranged in the same order of the DNA in the chromosome. Yeah. One chromosome of each homologous pair. The paternal chromosome comes from the organism's male parent, and the maternal chromosomes comes from the female parent. All right, and on the side here, we have the basic structure of meiosis. We have prophase 1, metaphase 1, and then metaphase 2, and then gametes. There's all the other interactions between that, but we'll go over it in a minute. Within the chromosomes, we have what's called alleles. The version of each gene is called alleles. Okay, so what is a gene? A gene specifically is a section of the chromosome that counts for each protein. All right, so each version of each gene is called an allele. So each allele is present in members of each homologous pair, and they may be the same or they may be different. Okay, and this is representative of either dominant or recessive. For a gene that encodes a protein, each different allele might encode a different version of that protein or no protein at all. So you'll have a dominant allele, you'll have a recessive allele, or you can have a dominant allele and what's called a zero allele. Each individual has a unique combination of these alleles in each homologous pair. Mixing mechanisms of meiosis and fertilization give each individual his or her unique combination of inherited traits. So you have the homologous pair, and you have one version of allele on one homologous pair, and then you'll have another version of allele on another homologous pair. They're both the same gene, is just a different allele. Or it could be the same allele, it could be, it could be the same or it could be different. Meiosis is a two-part process of cell division. Okay, the first part kind of duplicates the chromosomes, or duplicates the cell, and then the second part splits the cells. The duplicated chromosomes in the parental cell are distributed into 4 daughter cells, each with half the number of chromosomes of the parental cell. All right, so if you have a diploid cell, at the beginning you're going to have a haploid cell at the end. During meiosis 1, homologous chromosomes pair and non-sister chromatids undergo a physical exchange of chromosome segments. This is called crossing over. All right, so what happens during this first meiotic interchange is that the chromosomes will actually go through and physically exchange parts. All right, and I'll show you kind of how that happens in 2 slides from now. Completion of meiosis 1 produces 2 cells, each with half the diploid number of chromosomes. All right, so if you have diploid, what happens is they'll duplicate, okay? And so they'll have each 2 and 2. All right, so they'll have both diploid cells, still consisting of 2 chromatids. In meiosis 2, sister chromatids separate. Daughter chromosomes segregate into 4 different cells, each with haploid number of chromosomes. So at the beginning you have 2, it'll duplicate, those will split, and then you'll have 4 cells, each with haploid. And you can see this on the side here, how those differentiate. As your attendants, what I want you to do is tell me how this differs, how meiosis differs between mitosis. So let's go into meiosis. The first phase is prophase 1. Replicated chromosomes, sister chromatids, fold and condense in the nucleus. So at this point, you have the 2, and then it's duplicated. The 2 chromosomes of each homologous pair undergo pairing, and they form the tetrads. Right, those are those tetrads meaning 4. Chromatids and homologous chromosomes physically exchange segments. This is where they're exchanging those segments, and this is called crossing over. Sites where crossing over has occurred are marked by thickening spots, right? Chiasma. So those thickening spots, those spots where they're crossing over, it's just a thickening area. You can call it crossing over, you can call it chiasma. So what's happening is they're becoming so intertwined that the molecules are actually exchanging segments. And this is actually important for genetic diversity. Okay, remember what I said about alleles? So those alleles can exchange between those 2 chromosomes. And when you get down to the end, when you get down to those cells, when you get down to the 4 cells at the end, you can see the differences between those 4 cells. So one cell might have a different type of allele present than what it once was before, if that crossing over hadn't have taken place. So let's talk a little bit more into this chiasma, or this crossing over event. So we know that it's an exchange of chromosome segments. If there are genetic differences between these homologs, if there are some type of allele differences between these homologous chromosomes, you can produce new allele combinations. So whenever you're crossing over, if you have that allele difference between those two, you can produce this new allele combination that can produce a genetic difference in the gamete at the end of meiosis. So at the end of telophase 2, two nuclei receive an unchanged chromosome. So the two that haven't crossed over will be unchanged. And then the two receive changed chromosomes, and these are called recombinant chromosomes. So the two that have crossed over, those are called recombinant chromosomes. And they're called recombinant because they've combined and then recombined together to make a different pair of allele segments, or a different pair of DNA segments. They've recombined. Crossing over is a mechanism for genetic recombination. One or more crossing over events may occur in the same type of chromosome pair. So you can have one, or you can have multiple different ones. So all of that is occurring during prophase 1, right? During the time that it's pairing up, during the time that those forming tetrads are pairing up. Okay, once we get past that point, we can get into prometaphase 1. So prometaphase 1, the nuclear envelope starts breaking down, and once it breaks down, the spindle fibers can enter that nuclear area and then start combining and attaching to that kinetochore. Remember, we talked about that during mitosis. The spindle chromatids remain attached to each other. And then we get into metaphase 1. Metaphase 1, the spindle microtubules align the tetrads into that plane, into that center. Remember when we get into the center, if it's not in the center, sometimes it'll pull in one direction, or it'll pull more chromosomes into the other direction. It wants to get into the center so that it can pull them equally, separately apart. One of each chromosome pair is randomly attached to the spindle of each pole. Okay, so what happens here is it wants to make sure that they're equally separated. And this is a process known as independent assortment. Okay, so if it's all together in one set, it'll be independently assorted. There won't be any type of differences between one set and the other. And I'll talk about that more in detail when we get into genetics. But you should know that word. You should know independent assortment. You should know the law, the theory, the law of independent assortment. Anaphase 1 is next. The two chromosomes of each homologous pair separate and move to opposite poles as the spindle microtubules contract. So we know this. Once it's together, it can pull those homologous pairs to the opposite corners. Each chromosome still has the two sister chromatids, right? It's still diploid at this point. So it's still able to split those two homologous chromatids apart. Okay, telophase 1, this is a brief stage in which there is little to no change in the chromosomes, right? Telophase 1 is followed by what's called interkinesis, where the single spindle of the first meiotic division just disassembles and microtubules reassemble into new spindles for the second division. So interkinesis, there's a cleavage point in between the two cells, okay? Or if it's a plant cell, there's a plate that forms in between the two cells. It's called interkinesis because it's between two meiotic phases. Then we get into prophase 2. Prophase 2, chromosomes condense, and then the spindle fibers start forming again. So we have the two chromosomes that have the sister chromatids in them. Spindle fibers start forming up together. Prometaphase 2, the nuclear envelope breaks down again, or if it didn't break down the first time, it won't be formed up. Spindle fibers enter the nuclear area, attached to the kinetochores again. Metaphase 2, spindle fibers align the chromosomes in the metaphase plate, bring them to the center again. Anaphase 2, this time each chromosome pulls those sister chromatids apart, or each spindle fiber pulls those sister chromatids apart, separating them into haploid chromosomes. So the chromatids are segregated, right? Separating them into haploid chromosomes. Telophase 2, chromatids decondense, extend interphase state, spindles disassemble, and new nuclear envelope starts to form around the chromatin, and the result is 4 haploid cells. And you have some cytokinesis that's cleavaging all of those cells apart. So at the end, you have all these 1 haploid cells that are forming up those gametes. At some points, we can have an error in meiosis. Okay, so sometimes what's called non-disjunction can cause an error in meiosis. So in non-disjunction, the spindle fails to separate the homologous chromosomes in meiosis 1 or the sister chromatids in meiosis 2. In meiosis 1, what can happen is you get one cell that pulls a copy from all of the copies of one chromosome, right? So that other cell doesn't have the copies of all the chromosomes that it needs. So by the end, you have basically half the number of chromosomes that you need, and then the other half of your cells have more than the amount that you need in your gametes. So it's not functional. In meiosis 2, in non-disjunction, if you have the zygotes, if they receive an extra chromosome, basically one cell, one gamete, will have only one set of that chromosome. It'll have half the number of chromosomes that it needs. One cell will have basically 1.5 the number of chromosomes that it needs, and then the other two will remain fine. So this is just a weird it's an abnormal way of trying to make gametes. So if the spindle fibers don't do their job right, if the spindle fibers don't connect right, this is what happens. This is called non-disjunction. So in our bodies, the sex chromosomes, the pairs of chromosomes that make us different from the male and female side is the 23rd chromosome, right? So in females, it's XX. In males, it's XY, normally. So in meiosis, an X chromosome from the mother is able to pair with either an X or a Y from the father. A gamete formed in the female may receive either member of an X pair. A gamete formed from the male either receives an X or the Y chromosome. But that's not always the case in all animals, right? So in chickens, it's the exact opposite. So in chickens, the males carry a homologous pair, and the females carry the heterozygous pair. In insects, you actually have the females carrying the homologous pair, and the males either can carry a heterozygous pair or a what's called a haploid pair. Basically a haploid cell. So we talked about genetic variability when it comes to those crossing over events. Sexual reproduction's main benefit to evolution is its significance to genetic variability. So as reproduction is involved, it is imperative to genetic variability. Variability increases the chance that at least some of the offspring will survive in the changing environment. Basically, if you have a number of offspring that has some type of genetic variability, if you have a change in the environment, some of that offspring will survive to reproduce themselves. During meiosis and fertilization, genetic variability arises from 3 different sources. You have crossing over. That crossing over event paired between homologous chromosomes. You have independent assortment of the chromosomes segregated to poles during anaphase 1. And then the particular sets of male and female gametes that unite during fertilization. Okay, so it's not just, all of them going in at once. It's the random set of the sperm and the egg meeting together at once. All right, let's talk about some life cycles. So the animal life cycle is pretty well known. We know how the animal life cycle works. Time and place at which meiosis occurs follows 3 major patterns. The differences reflect the portions of the life cycle spent in the haploid and diploid phases. So all life cycles have a haploid phase and all life cycles have a diploid phase, right? So our haploid phase is usually the sperm and egg cells. Diploid phase is usually, the somatic cells, all the rest of the cells involved. In animals, the diploid phase dominates the life cycle. So we ourselves are the dominant life cycles. The haploid phase is reduced. The meiosis is followed directly by gamete formation. Right, so you have meiosis produces sperm and egg. Once those fertilize, they produce the zygote. Zygote makes the animal. This is different in plants. Okay, all plants alternate between haploid and diploid generations, and some algae and fungi. Depending on the organism, either generation may be dominant in the life cycle. So, usually earlier plants, earlier in evolutionary life, the generation is actually gametophyte. They actually have a haploid dominant life cycle. So fertilization in the diploid generation is actually reduced. Later in life, with trees and flowers and other things like that, those are produced in the diploid generation, and the more dominant generation is the sporophyte generation. Okay. Certain cells of mature sporophytes undergo meiosis. They produce haploid spores. They'll produce some type of spore that has a pollen grain in it. And that pollen will have spermatogenous tissue that will produce sperm, right? And that will land on a flower head or a cone head or something. And then that'll go into a ovary that has eggs inside of it. The gametophytes produce haploid eggs, and then the sperm will swim down into the egg and then fertilize it, and it'll make a zygote inside. And seed producing plants will have that egg inside and produce a zygote, and then it'll grow into a new plant. Life cycles in meiosis, kind of part 3. In some fungi and algae, the diploid phase is limited into a single cell. So this is kind of what I was talking about before. This is where the diploid phase is kind of reduced. The haploid phase is more dominant here. So the zygote is produced by fertilization. Immediately after fertilization, the diploid zygote undergoes meiosis to produce another haploid phase. Mitotic division occurs only in the haploid phase. So you have some type of spore that comes out and then goes straight back into a haploid phase. Haploid spores germinate to produce haploid individuals, what's called a gametophyte, which grow by mitotic divisions. Gametophytes form positive and negative gametophytes. That's basically what they call the male and female gender. The positive and negative gametophytes differentiation between the cells produced by mitotic divisions. Because gametes are produced by mitosis, all the gametes of an individual are genetically identical. This isn't really so-called the male and female portion. It's called the positive and negative because it's all undergoing through haploid phases. Once it gets into the zygote, that's when it's the positive and negative portion. Coming up on Monday, we have the exam. Okay, so I would suggest studying for it. I'm going to put up some video of a review for it soon, so make sure to look out for that. Be sure to go over that. I'm not going to go over a whole lot of questions. Just make sure to go over your notes, go over the PowerPoint slides and the previous videos that I've gone over, and make sure that you study. And I will talk to you later.